Okay, I see the virtual webinar room filling up fast. We're going to give it about 30 seconds to a minute, let everybody get settled in, and then we'll go ahead and get started. Indeed, it looks like we're going to have a good turnout today. Give it just a few more seconds. Okay, looks like it's leveled out. I think we'll go ahead and get started. Good afternoon, good evening, and good morning, everyone, depending on where you may be logging in from. I am Rob Burgess, and I head up business development for Sino-Biological. And I'd like to officially welcome you to the next installment in our BioTalk Tuesdays webinar series, where we have leading cutting-edge researchers talking about cutting-edge technologies in their respective fields. We have an excellent topic and an excellent speaker today who's focused on carbohydrate genomic regulation as it pertains to human disease. I will introduce the speaker to you and we'll get started in just a minute. I did want to mention one housekeeping issue, and that is if you could please type into the chat box, just please say hello and tell us where you're from. We're always interested in where people are chiming in from around the world. And also, if you have questions for our speaker, please just type those into the chat box, and we will get to those questions at the end of the seminar, and I will walk through them verbally and try to get to as many questions as I can with the speaker. And so with that, it is my pleasure and honor to introduce to everyone Dr. Ryan Weiss. Dr. Weiss is an assistant professor at the Complex Carbohydrate Research Center at the University of Georgia in Athens. He started this role in January of 2021. Dr. Weiss received his BS in chemistry in 2008 from Point Loma Nazarene University in San Diego, and his doctorate in chemistry in 2015 from the University of California, San Diego, under the supervision of Professor Yitzhak Tor. He then moved on to the Department of Cellular and Molecular Medicine at UCSD, where he worked as a postdoctoral fellow in Professor Jeffrey Esco's group. That's one of my own haunts. The CMM West is actually where I did my postdoc as well. So we've got something in common there, Dr. Weiss. Dr. Weiss's research program is focused on understanding the structure, function, and regulation of complex carbohydrates in human biology and disease. Additionally, his lab is dedicated to developing pharmacological and cell-based tools to aid in the discovery of novel therapeutic approaches for the treatment of congenital disorders involving glycosylation. And the title of Dr. Weiss's talk is The Genome-wide Regulation of Complex Carbohydrates in Human Disease. And Dr. Weiss, we will now turn the screen over to you, and thanks for joining us today.
Yeah, thank you so much for having me. It's really a pleasure to be here, and thank you, Rob, Priscilla, Amanda, everyone that was involved in helping getting this set up. So I'm going to share my screen. Hopefully we can make sure that works. Let's see. How does that look? Does that look okay? Okay, great. Awesome. All right. Well, thank you again for having me. Again, my name is Ryan Weiss, and my lab is located at the Complex Carbohydrate Research Center, which is shown here up at the top. This is at the University of Georgia, and my lab is located here on the 3rd floor, actually, so right here in this picture. And yeah, it's been a joy starting my lab about 3.5 years ago, and I'm really excited to share with you some of the work that my lab's been doing on trying to understand how these very complex and heterogeneous molecules, polysaccharides that are produced in our cells, how their biosynthesis is regulated and how it may be disrupted in different human disorders, and how we can use our knowledge of the regulation of complex carbohydrates to maybe think about how we can treat different human disorders. And so I just wanted to give this as kind of an overview slide. So my lab is a glycobiology lab, so we study complex carbohydrates, but we utilize a lot of biochemical, molecular biology, and genomic techniques to really try to understand how these polysaccharides are regulated at the genomic, epigenetic level, and how we can actually use this knowledge to treat different diseases. So just to give you a broad overview, we study some rare genetic disorders where these patients either can't break down or produce complex carbohydrates. And then, of course, cancer, tumor cells utilize these complex carbohydrates to help them proliferate and metastasize, et cetera. So just wanted to give some context to some of the work that I'll be talking about today. And this is the group. We need to get an even newer photo of the group, but these are the amazing postdocs, graduate students, undergraduates that work in the lab that really drive the lab and do all the work. So I'll have another slide at the end, but really thankful for them and helping, really kickstart the lab and really drive everything that we do. So for the non-glycobiology audience, I really like to start with this slide and to really put the work in the context of the cell. So all animal cells are surrounded by glycans, and not just animal cells, pathogens and prokaryotes and many other organisms utilize complex carbohydrates, and a lot of them do present it on the cell surface. So this is a human lymphocyte electron micrograph, and you can see this kind of fuzzy dark coating that surrounds that's above the plasma membrane of the cell. And this whole layer is collectively called the glycocalyx, but it's really a layer that's rich in complex carbohydrates that are attached to different proteins and lipids, and now RNAs and many other molecules that are called glycoconjugates. So this glycocalyx layer not only serves as a protective coating of the cell, but you can imagine that anything that wants to interact with the cell, the first layer that is going to come into contact with is this glycocalyx layer. So these sugars are our main way of not only protecting the cell, but also a way that our cells can interact with the extracellular environment. I show this picture to say that also our connective tissue, for instance, are rich in complex carbohydrates, and it forms a key structural role in the connective tissue of the basement membrane as well. So let's zoom in on the cell surface, and this is from the most recent version of the Essentials of Glycobiology textbook, which is actually freely available online if you're interested in learning more about these molecules. But if we zoom in on the cell surface, this glycocalyx layer is actually very diverse, and it's diverse not only in the glycans but also in the conjugates, so the proteins and the lipids and the GPI anchored proteins that are found on the cell surface that are decorated with these different sugars. And in the world of glycobiology, we actually utilize these different shapes to dictate the different monosaccharides. So you're going to be seeing a lot of these shapes today, particularly for these long linear polysaccharides. So I just wanted to give a brief overview, just say that each shape is just an easier way that we show these different sugars that make up these polysaccharides. And so while all of these different types of sugars and glycoconjugates are essential for maintaining cell homeostasis, essential for life, we're going to be focusing today on a specific subclass of these molecules known as proteoglycans. So this long protein right here shown in green is what we would call a core protein, a proteoglycan, that carries these long linear polysaccharide chains. These are known collectively as glycosaminoglycans, or GAGs, we call them for short. So these long linear polysaccharides decorate these proteins. These polysaccharides are very highly negatively charged because they have these sulfate groups all over them. We'll talk a little bit more in a second about how these are biosynthesized in the cell. But I just wanted to give some cellular context and show that these molecules are decorating the cell surface of all cells. So today we're going to be focusing on a subclass of these molecules called heparan sulfate proteoglycans. So heparan sulfate are these long linear polysaccharides that are highly sulfated molecules that decorate transmembrane proteins, such as the syndicans, GPI anchor proteins known as glycans, and can also be found covalently linked to different extracellular matrix proteins that are found outside the cell in the extracellular space as well. So a little bit about the function of these molecules. So heparan sulfate, again, is just one subclass of these complex carbohydrates that's found in all of our cells. If you have a genetic mutation and do not biosynthesize heparan sulfate, it's embryonic lethal. So I just wanted to state that to say how important these molecules are for life and for cellular function. And so there's a huge list of the role of these molecules in our cells, and I'm just showing some of them here. But this is a nice pictorial representation showing these carbohydrate chains, heparan sulfate chains that are decorating proteins. One of the key roles of these molecules is to bind growth factors. So they form a ternary complex with a receptor, in this case, a FGF or a fibroblast growth factor receptor on the cell surface. They are required to allow this growth factor to bind the receptor and then leads to downstream cell signaling events. So heparan sulfate is really important for growth factor signaling complexes and signaling events in the cell. They're also involved in cell-to-cell crosstalk. They can present chemokines and other ligands on the cell surface. There's also a lot of macromolecules that can bind to these sugars and get uptaken into the cell via endocytosis and other mechanisms. And they also play a structural role and are a key component of the extracellular matrix as well, and can be important for linking the ECM to the cell and allowing cell adhesion to occur and other processes. So, I've talked a little bit about heparan sulfate's normal roles in cells, but it also can be disrupted and be hijacked in various human disorders and other pathophysiological conditions. So I'm just showing a couple examples here. So tumor cells can actually upregulate proteoglycans and heparan sulfate on the cell surface, which can drive proliferation, angiogenesis, differentiation, etc. And a lot of pathogens, such as viruses, can actually bind to heparan sulfate as an attachment factor to our cells, which allows it, for instance, to interact with another receptor and then get uptaken into the cell. So this is a more recent example where SARS-CoV-2, which is the virus that causes COVID-19, was actually shown very recently in 2020 that heparan sulfate is a key attachment factor for SARS-CoV-2 to our cells. There's also neurodegenerative disorders, such as Parkinson's and Alzheimer's, where heparan sulfate plays a key role in the uptake, for instance, of tau and A beta. And if you look at plaques in the brain of many of patients with these types of disorders, heparan sulfate is rich in these plaques that form as well. And then the final example, and one half of my lab is really dedicated to studying rare genetic diseases where these patients are unable to biosynthesize or metabolize or catabolize these molecules in cells. And that can lead to, for instance, a rare skeletal disorder. This is called multiple hereditary exostoses. These patients have a genetic mutation where they cannot make it. They have a defect in the production of heparan sulfate, and it leads to these bony outgrowths called exostoses. As you can imagine, very painful, and it leads to chronic pain and many other issues in these patients. Then there's a whole suite of lysosomal storage disorders where these patients are unable to catabolize heparan sulfate in the lysosomes of their cells, and this can lead to developmental disorders, developmental delays, and major effects on the central nervous system and early death in these patients. So I just wanted to give a snapshot that heparan sulfate is not only involved in normal cell function, but it can be really disrupted in many disease states as well. So a little bit more about heparan sulfate. I think we need to talk about how it's made in order to understand some of the projects that I'll be talking about today. But heparan sulfate is biosynthesized in the ER and the Golgi of the cell. So the core protein is folded in the ER and then gets transited to the Golgi, where there's a whole host of enzymes that can covalently attach these sugar residues to serines of the core protein. And so I'm just showing some of these enzymes, and this is initiated by the addition of a xylose sugar by these two enzymes, XYLT1 and 2. There is then a specific enzyme that adds each of these sugar subunits, and this builds what's called a tetrasaccharide linker region. Then you get addition of this N-acetyl glucosamine sugar, and then polymerization of the chain by this EXT1, EXT2 complex. And just wanted to mention that rare skeletal disorder I just mentioned, multiple hereditary exostoses, is caused by mutations in one of these two genes. So the heparan sulfate is not able to polymerize. You get shorter chains, and it leads to these benign tumors that form on long bones of these patients. So after the sugar chain is polymerized, there's a bunch of different enzymes that will come and add sulfate groups to different positions. And why is this important? Well, if we zoom out, this is really what these heparan sulfate chains, they're long, linear, heterogeneous chains where there's some regions that are highly sulfated and some regions that are not very highly sulfated. And it's really the sulfated domains that really drive the biology or the function of these molecules. So for instance, there are proteins that can selectively bind to specific regions of the heparan sulfate chain. And one example of a protein that binds very specifically to heparan sulfate to this very specific pentasaccharide region is a protein, a serine protease inhibitor called antithrombin. So this is a protein that's found in the bloodstream. When it binds to heparan sulfate, or if you've heard of the anticoagulant drug heparin, it's just a highly sulfated form of heparan sulfate. It binds, these chains are allowed, they bind to antithrombin, cause a conformational change, and now this protein is able to inhibit the blood coagulation cascade. So I just wanted to give this as a highlight that there are these very selective interactions between protein ligands and these polysaccharides. Then there's growth factors and receptors that can bind to sulfation patterns of heparan sulfate. And so this is, I'm just showing fibroblast growth factors and the receptors. This growth factor may really like to bind to these 2-O-sulfated regions of heparan sulfate. So just when we zoom out, I just want to show you that the structure of these molecules really dictates their function in cell biology. And so if you have changes in the structure, it can really drive different binding events that can really have a major effect on cell biology. And so this is a final example of a molecule that might bind across these sulfated and non-sulfated domains as well. So where we're at now is this molecule has been studied for decades, and what we know is a lot about how these molecules are biosynthesized. So we know a lot about what enzymes are involved, where they are. They're in the Golgi, different compartments of the Golgi, and they add these different sugar or sulfate groups. But what we don't really understand is what regulates the production of these enzymes themselves. So, if you look across different tissues and cell types, some of these enzymes are expressed, some are not. And the expression of these enzymes really drives what the final structure of these molecules will actually look like. And so my lab is really interested in trying to understand what is controlling the expression of these enzymes and what controls their activity in the Golgi. We already know, for instance, that these two enzymes actually interact with each other in their obligate heterodimeric complex. Are there other enzymes that may be interacting and talking to each other and playing a key role in really driving the biosynthesis of these molecules? And is this a temporal, spatio-temporal, cell-specific effect or not? And so this is kind of what I'm going to be talking about today. And this is just some data from papers from quite a while, but about 20 years ago, that showed that for instance, if you isolate heparan sulfate from different mouse tissues, there's a huge diversity in the amount of heparan sulfate that's produced in these different tissues. And in fact, if you look at RNA sequencing, so this is from the Protein Atlas database, this is from human tissues here shown on the top. And this is looking at even the gene expression of the different enzymes that are involved in making heparan sulfate. You can see that there's a huge diversity in the expression level of some of these enzymes. While some of these are very constitutively expressed across different tissues, others are only highly expressed, for instance, in the brain. And so we really don't, in the realm of glycobiology, of studying complex carbohydrates, we really don't understand what controls and regulates the expression pattern of these enzymes and what leads to this huge diversity in not only the amount, but also the composition of these heparan sulfate chains as well, which again, the structure really drives the function of these molecules. And so this is just an example of showing that heparan sulfate expression actually can be very perturbed and altered, for instance, in disease states versus normal tissue. So this is gene expression from different human cancers. And you can see that across these different cancers, there's a huge diversity in the expression level where the changes in expression of these different enzymes in tumor versus normal. And really what's interesting is if you break this down, you can see that most of these changes are actually in the enzymes that add sulfate groups that really alter the fine structure of these chains, which drives these ligand and protein interactions at the cell surface. So you could, I really want to highlight that the regulation of these complex carbohydrates is a lot more complex because these molecules are not made by a template. So, our classic central dogma of biology is that DNA is, transcribed to RNA, translated to protein. And so this is really dictated on the DNA level, right? And then there's regulatory mechanisms along the way as well. But in terms of complex carbohydrate biosynthesis, this is a very non-template driven process, meaning that you have all these monosaccharide donors sitting around in the cell or in the Golgi, for instance. Then you have these enzymes that can add them. But how is this whole process controlled? Because if you look at different cell types, you see very distinct patterns of glycans that are found in these different cell types or in a disease state versus normal. And so one major part of goal of my lab is to really understand how this process works. So you can imagine before these molecules are presented at the cell surface, there's multiple steps that can be regulated along the way. So you could have regulation in the nucleus, where you have regulation of the expression of either the core protein, shown in green here, or the biosynthetic enzymes that are involved in constructing these chains. This could be epigenetic or transcriptional regulation. Then you could look in the ER and the Golgi. There may be chaperone proteins and scaffolding proteins that might alter how the biosynthetic enzymes are interacting with each other or their activity. There could be regulation of the substrate or the pools of molecules that are needed, for instance, to sulfate these molecules. There could be regulation on the transport or the recycling or the N-cytosis of these molecules. There could be factors that are outside the cell. We already know of a couple examples, which I'm going to be talking about today, of factors that can come and cleave off, for instance, specific sulfate groups and really drive the interactions of these molecules at the cell surface. So my lab is taking different strategies to identify novel regulatory factors. And I'm going to be focusing mostly on looking across the genome and using tools such as genome-wide CRISPR screening, and also harvesting and mining data that's already out there, genomics data, to maybe point us in the right direction in terms of finding factors that might be regulating the expression of these enzymes. Then we have a whole other part of the lab that don't have time to talk about today, but is to utilize these findings, but also finding drugs that might be able to tune the biosynthesis of these molecules in disease. So the first story I'm going to be talking about today is utilizing genome-wide CRISPR-Cas9 screening approaches to really take unbiased screening approach to identify factors that regulate the production of these molecules in cells. So hopefully everyone knows what CRISPR is and how this works. I'll just talk briefly about this. So CRISPR was really came to the forefront about 10 years ago, and it was discovered that this system was originally found in bacteria as a defense mechanism, for instance, against invading viruses. But some really amazing labs and amazing work by Feng Zhang and Jennifer Doudna and many others found that you could actually repurpose the system into mammalian cells, for instance, and you can actually target and knock out whatever gene you want by selecting a guide RNA that will then target this protein, a nuclease called Cas9, to whatever site in the genome that you want, and it cause a double-strand break and cause, for instance, a mutation and lead to, for instance, a knockout of a given gene. And so there's been really amazing strides to take this to a genome-wide level. You can actually purchase these guide RNA libraries, for instance, from Adgene. And so we utilize this guide RNA library called the Gecko library that targets about 19,000 genes in the human genome. The library includes over 122,000 different guide RNAs that target all these different genes. And we have 6 guide RNAs per gene for redundancy to ensure that we can target all these 19,000 genes. And so you can purchase this library, make a lentiviral version of this library in HEC293 T cells, and then you can infect the library into whatever cell type you're interested in. And so we used a human melanoma cell line that expresses heparan sulfate in the cell surface and has been utilized in these types of screening approaches, especially early on. And so what you can do is you can stably express the Cas9 nucleus in these cells and now transduce them with this library at a very low MOI to ensure that each cell has a different gene that is targeted. And so as a result, you end up with this library of millions of cells where each cell has a different gene that's been knocked out with the CRISPR-Cas9 system. And so this was done in collaboration with Nathan Lewis at UCSD and a postdoc in his lab as well. And so what's really powerful is once you have this library of cells, you can ask very big biological questions. So our question was, what regulates the production of heparan sulfate in these cells? And so to do this, we had to think about a screening strategy where we could detect heparan sulfate in the cell surface in a very robust way, in a way that we could detect either through flow cytometry-based methods, which I'm going to explain here on the right, or in a cell killing type of assay. And so we actually utilize a small molecule approach. So this is a molecule called guanidinoated neomycin that was actually originally discovered to bind to DNA by Yitzhak Tor at UCSD. And his group ended up discovering that outside the cell, when you add this to cells, this molecule, which is really just a neomycin antibiotic where all the amine groups have been converted to guanidinium groups, this molecule is highly positively charged now, and it actually binds selectively to heparan sulfate on the cell surface. And so what's even cooler about this molecule is that you can actually attach large cargo to it, and it will bind selectively to heparan sulfate in the cell surface and get endocytosed into the cell. You can actually deliver large cargo through a heparan sulfate mediated uptake mechanism into mammalian cells. And so we wanted, as a proof of concept, what we did is we attached a large cargo, in this case, a saporin toxin that does not get into cells by itself. But when you conjugate it to Geneo, now you can dose dependently kill cells. And these are wild type A375 cells. And what we wanted to show is that we knock out, for instance, a key enzyme involved in sulfation of heparan sulfate. You get this large shift in resistance to this toxin conjugate. And if you knock out all sulfation altogether, so this is the PAP, an enzyme that's the PAPS transporter transports PAPS, which is a sulfate donor, into the Golgi. So in this case, your heparan sulfate molecules and many other molecules are not sulfated at all. You get even greater resistance to this toxin. So we could utilize this in a screening approach where we could take our library of cells that have a different gene knocked out in each cell and subject this library to a geneosaporin treatment. And we could also treat with PBS as a negative control. And what you end up with are cells that become resistant to the toxin. And we know that most of these cells may likely have defects in heparan sulfate biosynthesis. So we can collect these cell populations and isolate the genomic DNA and PCR amplify the stably integrated guide RNA. Because again, we use a lentivirus to introduce this guide RNA library. And then we can use next generation sequencing to identify which guide RNAs are highly enriched in these resistant cells compared to our general cell population. And in parallel, we utilize a slightly different approach where we use Geneo, but now we conjugated a dye to this. And we did this assay on ice where we could actually just look at binding of Geneo to the cell surface, and then we can sort cells using flow cytometry. What we were looking for is for cells that had a decrease in binding to this Geneo-Cy5 conjugate. And again, we can isolate the genomic DNA and subject the PCR products to next generation sequencing. I want to highlight that for those of you interested in doing these types of screens and maybe do not have expertise in bioinformatics and coding, there's a really easy to use web-based tool that we developed in collaboration with Nathan Lewis's group called Pineapple Pi. So this is a website where you can drag and drop your sequencing files, and it allows you to do all these analysis of this next generation sequencing of one day. So it's a really powerful way to be able to analyze these types of assays. So the first question we wanted to ask is, okay, if we treat ourselves with these Geneo-toxin or the Geneo-Cy5 conjugate, do we identify top enriched genes that we know are involved in heparan sulfate biosynthesis. And sure enough, the majority of the genes that we found enriched in the toxin resistance screen, as well as our FACS sorting screen, were all known enzymes or proteoglycans that are known to carry heparan sulfate molecules at the cell surface. And so to orient you to these graphs, this is fold enrichment, or the amount of counts that we had in our resistance fraction compared to our total population. And over here is from our cell sorting experiment, the ones that had low genome binding versus the ones that had normal genome binding. So this is a really great positive control. And so next, we wanted to investigate whether do we get novel factors, and this is really what we're interested in, is do we have factors that have previously not been implicated in heparan sulfate assembly? And so we did, sure enough, find a ton of other pathways enriched. And these were the top pathways, of course, heparan sulfate itself. Then we had genes involved in chromatin remodeling factors, we had growth factor signaling pathway molecules, GTPase, protein modifiers, and transcription factors as well. And so we originally published this factor KDM2B, which is a chromatin remodeling factor, histone demethylase, which was a novel factor that was found to be involved in fine-tuning the structure of heparan sulfate via epigenetic regulation of these enzymes. And so we're not going to focus on that today. I'm going to focus on a more recent findings. So a project that Dr. Amrita Basu is a very talented postdoc in my lab. I took on this project when we first started my lab. And so we are investigating this transcription factor TFCP2, which was a top hit in this CRISPR screen. The TFCP2 is a CB2 transcription factor. It binds directly to DNA, where it can regulate the expression of many genes involved in cell proliferation, angiogenesis. It's been genes implicated in tumor metastasis, EMT, etc. It can also act as a co-regulatory factor, where it can, for instance, interact with YAP and then regulate transcription factors and downstream genes in that manner as well. And this is an example, for instance, in hepatocellular carcinoma. And this is a plot showing the primary binding site based on ChIP-seq experiments for this transcription factor. So this has been studied a lot in the context of cancer, where it's typically upregulated, for instance, in hepatocellular carcinoma and other types of cancers as well. And so we were interested in asking this question of, does TFCP2 regulate heparan sulfate assembly? And could it be that overexpression or altered expression in this transcription factor may drive changes in glycosylation or in these complex carbohydrates, which may drive tumor genesis, for instance. So again, we worked in our human melanoma cells. TFCP2 is interestingly upregulated in melanoma as well. So we're using this as our model system. So we could use CRISPR and individually knock out TFCP2. And the first thing we asked is, are there changes in heparan sulfate assembly? And then does those changes in structure impact the function of these molecules? And so we have some really cool liquid chromatography and mass spectrometry techniques where we can purify these highly negatively charged molecules from cells, from the cell surface or in the secreted media. This is on the cell surface in this instance. And we can actually break down these sugars, these highly heterogeneous long polysaccharides, into their disaccharide subunits using lyase enzymes that are commercially available. This allows us to really ask the question of, what is the composition of these chains? Because currently we're still waiting for amazing, top-down approaches where we can literally look at the whole chain in and of itself. So nanopore technologies and others are being investigated currently, where it would be great if we could just feed these through a nanopore and really figure out what the actual structure of these intact chains are. But for now, what we have to do is break them down into their subunits. And so this is what the data actually looks like after we run it through the LC-MS. We use standards where we can actually absolutely quantify how much of each of these disaccharides are present. And so we can actually look at the composition. So how this works is we have different disaccharides going from non-sulfated disaccharides, something that would look like this, to something very highly sulfated, which would look like something like this that has 3 sulfate groups on it. what was really interesting is that most of the disaccharides didn't change except for this highly sulfated D2S6 disaccharide, which was significantly down in our TFCP2 knockout cells. Interestingly, the total amount of heparan sulfate didn't really change between the wild type and knockout. And this is just another way to show this data and show that it seems like these highly sulfated subunits are what are majorly affected in these TFCP2 knockout cells. So next we asked, okay, do these compositional changes actually lead to changes in function? And so we could do cell surface binding assays. As I mentioned earlier, heparan sulfate is essential for these growth factors to bind to the receptor on the cell surface and lead to downstream signaling events. So we can actually use isolated growth factors and treat cells and look at binding to heparan sulfate specifically on the cell surface. And so what we found is that when we add FGF1 or FGF2, these are two different growth factors that bind to different sulfation patterns in heparan sulfate. We found a significant decrease in FGF1, but interestingly, no change in FGF2 binding. We expanded our binding assays to look for antibodies that detect global heparan sulfate levels. This was unchanged. Global proteoglycan levels, this was unchanged as well. And global chondroitin sulfate, which is a related other type of glycosaminoglycan, and there was no changes in that as well. And we found a significant drop in antithrombin binding, which again is a protein that binds to this very specific pentosaccharide subunit in heparan sulfate and heparin. So these results suggested that when you knock out TFCP2, it doesn't result in global drop in heparan sulfate in the cell surface. Instead, it seems that the sulfation patterning may be altered, which really drives changes in binding of certain growth factors or other ligands. And what we showed is that if you transfect back in TFCP2, you get expression, it actually rescues this FGF1 phenotype, suggesting that it truly is the knockout of TFCP2 that's driving these changes in binding at the cell surface. So we took a global approach and did RNA sequencing to look for, since this is a transcriptional regulator, we wanted to ask globally, what kind of changes are happening to the transcriptome? And interestingly, you see that when you knock out TFCP2, many genes go up and some go down, suggesting that TFCP2 may play a role in not only transcriptional activation, but also repression. And what was really actually surprising is that when you do a gene ontology analysis of these significantly up or down regulated genes, the major pathway that was affected was the extracellular matrix. So these are genes involved in proteoglycans, collagens, fibronectin, and other extracellular matrix proteins, as well as enzymes that are involved in making these glycoconjugates as well. This is a bit surprising, but very interesting considering that we were looking at these molecules that are a key component of the extracellular matrix. And so when we zoomed further in to our RNA-seq data, we looked specifically at enzymes that we know are involved in heparan sulfate biosynthesis or the proteoglycans themselves. And what we found is that multiple genes were differentially expressed in our TFCP2 knockout cells. So this is on the y-axis, a log2 fold change in gene expression versus wild type. And then on the x-axis is the basal expression level. So something over here will be highly expressed in wild type, and then these would be the changes that we observed. And so we found changes in this SULF1 and SULF2, which are interestingly both secreted proteins that will bind to heparan sulfate and cleave these specific 6-O-sulfate groups, which really drives, for instance, FGF1 binding events. And in terms of the proteoglycans, we didn't see a lot of major changes, except this drop in this glycan-4 proteoglycan. So we wanted to look into this further and do rescue experiments where we genetically restored SULF1 back to wild type levels using siRNA. This restored FGF1 binding and even increased it above wild type a little bit. We tried to transfect back in this HS6SC2 enzyme, which adds this 6-O-sulfate group. Interestingly, this did not rescue the FGF1 binding phenotype. And then finally, we used this glycan-4 expression construct. And I think I have to mention here that these are all from Sinobiological. So thank you guys for making these expression constructs available. And it's just easier than having to clone all these ourselves. So we utilize these types of products a lot. And so we were able to show that even when you rescue these proteins, it didn't really, only SOLF1 was really driving the rescue of this FGF1 binding defect. And so we wanted to probe this further. And so we cloned the SOLF1 promoter in front of a nanoluciferase. This promoter has a TFCB2 binding motif. And what we showed is if we transfect this plasmid and look at luciferase expression in wild type versus the knockout cells, we had much higher expression of the nanoluciferase in the knockout cells, suggesting that TFCB2 may be a repressor of SOLF1 expression. And again, we're interested in SOLF1 because it's the more highly expressed SOLF in these cells. And again, this protein's function is to remove these 6-O-sulfate groups, which can really alter FGF1 binding, and that is really shown here in these rescue experiments. So as an orthogonal approach, we also looked at pharmacological inhibition of TFCB2, and so we use a small molecule where we can inhibit TFCB2 binding to DNA. So down here on the bottom right, you can see that this molecule does not affect TFCB2 protein levels, but it does block the binding of TFCB2 to its genomic targets. And so as a result, the higher dosing of this drug decreased FGF1 binding and increased SOLF1 expression, also showing that TFCB2 seems to play a role in SOLF1 expression. We also tried knockdown of TFCB2 in a host of different cells, for either other melanoma cells shown here on the right, or other cell types or other cancer cell lines. And what we showed is that we could, knocking down TFCB2, interestingly, really only decreased FGF1 binding in melanomas, suggesting that TFCB2 may have a specific effect in regulating FGF1 heparan sulfate interactions in melanoma compared to other cancer types. So we investigated this further, looking at melanoma cell growth, either in a clonogenic assay, 2D assay, or in 3D using anchorage-independent growth assay. And we showed in all cases that when you knock out TFCB2, there's a decrease in 2D growth as well as 3D growth, suggesting that TFCB2 plays a key role in cell growth and of these tumor cells. What was really exciting to see is that if we only restored SOLF1 back to wild type levels in our knockout cells, it restored the growth. This was a really exciting result because it suggested that heparan sulfate and its fine structure is really what's driving the growth of these cells. And so we actually turned to the TCGA database and looked at patient data from skin cutaneous melanoma patients. And what we saw is that high expression of TFCB2 actually leads to lower survival. And in the same way as our experiments show, that low expression of SOLF1, which is probably there when TFCB2 is there, also leads to lower survival as well. So it really suggests that TFCB2 is a repressor of SOLF1 expression, which really drives all these cellular phenotypes. And so we're interested in investigating this further and really understanding, how genomic regulation of heparan sulfate biosynthesis and its structure and function in tumor cells may play a key role, for instance, in melanoma survival, but also resistance mechanisms. This is from a recent paper that showed that actually when you treat with like a standard treatment of care, which would be a BRAF inhibitor, melanoma cells go into this senescent state and actually upregulate FGF1. This upregulation of FGF1 actually stimulates the growth and survival of these cells and leads to resistance mechanisms as well. So we're really wondering if heparan sulfate is playing a key role in this type of mechanism. And so hopefully I was able to show you in this first segment that genome-wide CRISPR screening, it really is a powerful approach to identify novel factors that you would never previously been implicated in heparan sulfate biosynthesis. And so we were focused on one of these factors, and TFCB2 seems to regulate the fine sulfation patterns of heparan sulfate, which regulates FGF1 binding, and downstream we're able to show that it really seems to impact melanoma cell growth as well. So in the last part of the talk today, I just wanted to focus a little bit on other approaches that we're taking to identify novel regulatory factors. And again, this is kind of a big black box in glycobiology. How are these non-template driven biosynthetic processes controlled at the cellular level? And so for this, we've turned to using different bioinformatic tools. And interestingly, these tools are named after Simpson characters. And so this Homer was originally developed at UCSD by Chris Brenner and others, and published back in 2010. So this is utilized a lot in genomics research to analyze large genomic data sets. And so in a paper that was published back in 2020, we showed that you could actually use this program, this software, and actually just ask, what if I just put a group of genes that are involved in the same biosynthetic process through Homer and ask which transcription factors may bind or have motifs in the promoter of these genes. And so this is a really powerful tool. Classically, it's used, you can put your RNA-seq data set in and say, okay, which genes are different, of these differentially expressed genes, which pathways are there. You can put ChIP-seq data in and say, what is the binding motif of my transcription factor that I pulled down? So we use it in a slightly different way and said, well, what if we just put all these genes in that are involved in heparan sulfate biosynthesis and say which transcriptional regulators may be able to bind to the promoter of these genes of interest as well. And so, okay, thank you. Yes. And so we're really excited to use this tool. And this is a paper that was published in 2020. We were able to identify this novel zinc finger transcription factor. It was highly enriched, this binding motif was highly enriched in the promoter of the genes involved in heparan sulfate assembly. And sure enough, we found that when you knock out this zinc finger, which previously, if you looked in PubMed, there was virtually nothing known about this factor except its binding motif, which is great for us because we went and knocked this out and asked unbiasedly, okay, if we knock this out, what happens to heparan sulfate assembly? What was really exciting to see is that if you knock this factor out, interestingly, antithrombin binding went through the roof. So antithrombin, again, is this protein that binds to this very specific pentasaccharide subunit. And what's really interesting about this is that this is highly relevant in heparin therapy, which is an anticoagulant drug that's given routinely all over the world to keep your blood from clotting. And so we were asked our question, we really dug into the data and showed that this zinc finger 263 transcription factor seems to repress the production of heparin. And so when you knock it out, you can really turn this on, this ability to bind to antithrombin. So we're really interested in investigating this further and thinking about what if we could bioengineer cells to make this anticoagulant drug heparin? So that's something we're investigating. So in this final little piece, I want to show you that we can use more updated versions of Homer. In this case, we use this software called BART, which stands for Binding Analysis of Regulation of Transcription. So the BART takes another whole layer to this, is that not only does it ask, it really utilizes ChIP-seq data sets, actual data, and says which factors actually bind to the promoter. Here on the left, this is just a prediction software to say, hey, there are potential binding motifs there for transcription factors that may be relevant. Here it takes actual data and it shows, okay, which factors are actually binding to your gene set of interest. And so we did this again with our heparan sulfate genes. We put this into BART, and what you're seeing is an enrichment score, which shows, okay, of all these ChIP-seq data sets, which factors are most widely found to be binding at the cis regulatory regions of your gene list of interest? And one of the top hits we found was this EZH2 protein, which is a histone methyltransferase. And I just want to highlight that what was really interesting in blue is we found multiple proteins that all form a complex called the polycomb repressive complex. And we also found other factors that we previously already know are involved in heparan sulfate assembly, which helped validate this search protocol that we used. And so we ended up targeting and looking into this top hit, which is EZH2, which is a histone methyltransferase. So this is a protein that adds a histone at lysine 27 of histone 3. It adds this trimethyl mark. which then recruits this other histone complex, PRC1, which then adds this ubiquitin group, which then leads to repression of chromatin and leads to gene regulation. And so EZH2 has been studied extensively, particularly in embryonic development and differentiation. It's also been found to be mutated and overexpressed in many different cancers, melanoma being one of those, where about 5% of melanomas have upregulation or a mutation in EZH2. And so we were interested in investigating if EZH2 could also be a regulator of heparan sulfate production. So again, we turned to disaccharide analysis, and this was work done by Neil Patel as a PhD student in my group. He was actually the first person that joined my lab back in 2001. And so we knocked out EZH2 in these cells, and sure enough, global histomethylation levels are gone. And what was really exciting is that we again found fine structural changes in heparan sulfate using our disaccharide analysis workflow. And what we showed is that we actually found an increase in FGF1 binding in these knockout cells. And again, without major changes in global levels of heparan sulfate. So we found this differential binding effect where we have an increase in FGF1 binding and a drop in antithrombin binding when you knock out EZH2. When you look at gene expression in these cells, you can imagine when you knock out EZH2, many genes will change in their expression, and that is true. And what we found is that selectively, it seems that the sulfatases again come up as being differentially regulated. So when you knock out EZH2, you get a drop in SULF1 expression, but an increase in SULF2. And you get a drop in some of these other enzymes that are involved in adding these different sulfate groups. And interestingly, when you zoom out, many of the major pathways that are affected in these cells when you knock out EZH2 are involved in glycan biosynthesis, and particularly heparan sulfate. So we showed pharmacologically that if you inhibit EZH2's activity, so you can see this drop in histomethylation, you again get an increase in FGF1 binding as well. So we were interested in asking, is this regulation through its canonical mechanism where it interacts with this PRC2 complex, which leads to gene repression and compaction of the chromatin? So we targeted two other subunits of the PRC2 complex with siRNAs, SUS12 and EED. Interestingly, we do not see changes in FGF1 binding in these cells. And sure enough, when you look at gene expression, the only one that we really saw increase is SULF2, suggesting that SULF2, if you knock down or knock out any of these subunits, you see an increase, suggesting that the PRC2 complex likely canonically represses expression of SULF2, which is really not, has not been known before. And then what's really interesting about EZH2, and I'm going to finish up shortly, is that EZH2 can actually function outside of the canonical PRC2 complex, where it can interact with other proteins and actually lead to gene activation as well. So we were interested in investigating this as well. EZH2 can interact with a lot of different proteins. So we used co-immunoprecipitation and proteomics to identify interactome of EZH2 in these cells. And sure enough, we found a lot of the canonical regulatory factors, and this was done in collaboration with Lance Wells here at the CCRC. But we found other factors that have been investigated previously. One of those was TRIM28. So this is another nuclear localized factor that, interestingly, has been shown previously to interact with EZH2. And we combed the literature, and what was really exciting to see is that this group that really does not study heparan sulfate or glycans, they showed that one of the top regulatory factors that was co-regulated by EZH2 and TRIM28 was SULF1. And I'll remind you that SULF1 was lowered in our EZH2 knockout cells. And again, this paper doesn't even mention heparan sulfate, even though this is one of the top factors that they found. TRIM28 can play a role in melanoma, where it actually is a switch between tumor growth and invasion as well. So we were really interested in asking if maybe this EZH2 TRIM28 complex may regulate SULF1. So we actually knocked down TRIM28 in these cells, and we saw a decrease in SULF1 expression, which is exactly what these prior studies showed in breast cancer cells. And so we investigated this further, and we showed that, again, if you knock down TRIM28, SULF1 goes down. What's really interesting is if you knock out EZH2 or knock down TRIM28 or just SOLF1 itself, they all lead to an increase in cell growth, suggesting there's something happening that's driving the growth of these cells when you target all these molecules. And this one I'll just highlight as selectively altering and really increasing heparan sulfation. So Neil has investigated this further. We've shown that if you target SOLF1, which is heparan sulfate specific, or TRIM28, or EZH2, it leads to an increase in clonogenic growth of these cells. But interestingly, we see differential changes in cell migration. And we've looked in the TCGA database, and interestingly, in metastatic tumors from cutaneous melanoma patients, there's an increase in SOLF1 and TRIM28 in the metastatic tumors. So we're really interested in wondering if EZH2, TRIM28, this regulatory axis may be driving the switch between localized tumor growth and metastasis. So we need to do the in vivo models in mice to really get at this question. And final slide, I want to show that we're also working on doing chip sequencing, which is really asking the question is, does EZH2 localize differentially or with or without the PRC2 complex at these different genes, for instance? So this is the SOLF1 locus or SOLF2, and I just want to draw your attention that SOLF2 is very lowly expressed in these wild type A375 cells. You can see a huge amount of the canonical PRC2 complex there, which leads to histomethylation and gene repression. But interestingly, if you look at the SOLF1 locus, this gene is more highly expressed in these cells. EZH2 and TRIM28 seem to co-localize in the cis-regulatory region of SOLF1. So we're investigating whether EZH2 or TRIM28 really help the other one localize to the cis-regulatory region and regulate SOLF expression. So hopefully I was able to show you that we're able to also mine different databases to really look into the different regulatory role of these epigenetic factors, which again, we don't really know much about in the realm of glycobiology. So with that, I want to thank all the funders. This is a T32 glycoscience training grant that helped fund Neil and another student in my lab, Megan, NIH. Neil recently received this ARC Scholar Award, which I'm proud of him for. And I want to thank all our collaborators and thank you all for listening. And sorry if I went a little bit over time, but be happy to answer any questions.
Thank you, Dr. Weiss. Fantastic seminar. We certainly appreciate it. It's very enlightening. We have time for one question, and we've got a good one here in the chat box from Jesse Chen. Jesse is asking the question, in the CRISPR screening, how did you validate that the multiplicity of infection is low yet sufficient enough to generate just a single gene knockout in each cell?
Yeah, that's a great question. So this is a statistical estimation that we do, but we do this, we validate the MOI functionally. So what I mean by that is that each of these plasmids that come in this library of 120,000 different guide RNAs, each plasmid also has a puromycin resistance cassette. And so what we do is when we first make our lentiviral library of this CRISPR guide RNA library, we functionally titer different amounts of the virus onto the cells, and then we select with puromycin. And what we're looking for is what amount of the cells actually survive after puromycin selection. So if I treat the cells with the virus and all of the cells survive, we know that you have at least an MOI of 1 or more. And that we do not want to do that because statistically speaking, you're most likely getting more than one viral particle per cell. And so what we do is we select a viral titer, a viral amount that we put on the cell based on a survival of about 30% of the cells. And so we usually shoot for an MOI, and this again is a functional MOI of about 0.3. And this is typically how it's done in the field. So statistically speaking, most likely the majority of the cells will only get one viral particle. It doesn't mean that every cell only gets one viral particle, but the majority of them likely will. Hopefully that answers your question.
Great. Thank you for that answer.
Yeah.
Sounds pretty convincing to me.
Yeah.
Isaac and Guma, sorry we weren't able to get to your question because we're kind of out of time, but we'll get it answered by email for you, Isaac from Nigeria.
Yes. Yes.
Go ahead and Yeah, and I'll go ahead and thank everybody, attendees. We had a great turnout today. We appreciate it. Just real quickly, we had folks from Atlanta, Oakland, Mexico, the UK, Pakistan, Morocco, Jamaica, Belgium, Sweden, and of course, Nigeria. And just a fantastic turnout. Dr. Weiss, I really want to thank you. Excellent seminar. Congrats to you and your team for all your hard work and all your success in what I think is a very difficult field.
No, thank you.
Really
No, thank you
fun.
so much for having me. It was great to be here. And yeah, again, if anyone would like to reach out, I'm sure that you guys have my email address, or I have a lab website as well if you Google, and happy to answer any follow-up questions that anyone may have. So yeah.
Thank you for that. And I also have to thank my colleagues, Priscilla Hernandez and Amanda Tamalka. They're the ones that actually executed the webinar today. And one more thing, there's a great football game going. Good luck to your Georgia Bulldogs this weekend. But I have to say, I went to UT and I live in Austin, so hook 'em horns.
Go Dogs, go Dogs. Hopefully it'll be a great game. So yeah.
Cheers, it'll be a good one. Thank you, Dr. Weiss. Everybody have a good day.
Yeah, thank you again. I really appreciate it. Take care.
Bye-bye.